Hi, I'm Hassan, the lecturer here at the Institute of Medical Education, and today we're going to go through a biology GAMSAT unit. It's going to be, around cell cycle, mitosis, and meiosis, and there's going to be two questions relating to the unit. So before we begin, I'll give you a chance to have a read of the stimulus. So you can pause the screen now and have a read of the stimulus here, and you can have a read of the first question, so question 75. So you can pause the screen. And now question 76. So you can pause the screen and have a watch or have a listen. Oh, sorry, have a read. And okay, so we can start now. So once you read the stimulus, it tells us that cyclin-dependent kinases, they're these sorts of, enzymes or proteins that regulate cellular activity and signals for replication and signals to induce mitosis and meiosis. So it's important to note that interphase is where the cell spends most of its life. So interphase precedes mitosis and meiosis. So when we read question 75, it says, at which point does the mitotic process first display positive feedback? So when we read the stimulus, we're told that these cyclin-dependent kinases, once they become phosphorylated and activated, there's a cyclin-dependent kinase at each stage of interphase, which can move the cell along the cell cycle. And then obviously there's going to be cyclin-dependent kinases for the mitosis phase as well. But it's important to note that the question is asking at which point does the mitotic process first display positive feedback? So positive feedback in this instance would be cells growing, and then the cyclin-dependent kinases are activated, and they kind of act like a gate to tell the cell, hey, let's start DNA synthesis, and then let's start cell growth. So there's this positive feedback happening once cell growth occurs, then synthesis. So DNA synthesis, then cell growth again, then mitosis and meiosis. So there's this positive growth that's occurring right from the beginning, from interphase all the way through to mitosis. And we're told this in the stimulus. So it's important to note then that if the question says, when is it first occurring, the positive feedback, it can't be mitosis because mitosis occurs after interphase. So remember the acronym IPMA. So interphase, then mitosis or meiosis, prophase, metaphase, anaphase, telophase. So that occurs after interphase. So that means it can't be mitosis. So it has to be one of these three. So we know the beginning of interphase, so it goes from G0 to G1, then DNA synthesis, then cell growth because it's preparing for mitosis. So you know straight away that it has to begin here with the cell growth where there hasn't been any DNA synthesis. So you know also that if there's going to be a signal to cause DNA synthesis, it has to occur between G1 and S. So therefore, if it's the first instance to display positive feedback, it has to be the answer G1 to S. So that's the answer here. Yes, you can ensure that the CDKs are upregulated throughout interphase and mitosis and meiosis, but the question isn't asking, well, is it occurring? It's asking when is it first displayed? And it's first displayed here from G1 to S phase. So if we move on to the second final question here, number 76, it's important to note first and foremost, the stimulus is talking about mitosis, but the stimulus here is talking about meiosis. So please differentiate between the two because we know that mitosis produces two exact daughter cells, so identical. And you can tell straight away this is meiosis because we've got four daughter cells of differing chromosomes. So these are different types of chromosomes here. So you get four daughter cells that are not identical as opposed to two identical daughter cells in mitosis. And it's important to note that in mitosis, we produce diploid cells, whereas in meiosis, we produce haploid cells. So one set of the chromosome, whereas in the diploid, two sets of chromosomes. So one from your mother, one from your father. So you usually find these meiotic cells or these zygotes. So it's sperm cells and the ovaries, the eggs in the females. So males. So if I get my pen out here, get my pen, we'll draw it. So diploid, it can be represented, you've probably seen it as 2N. Haploid is N. So this is just referring to, in humans, we have, 23 chromosomes. But remember, because you get one from your mother and your father, that's why we're diploid. So we're diploid organisms. So that's haploid. We have 46 chromosomes. That's why you often hear that humans have 23 pairs of chromosomes. So when we say diploid, you've got 2 pairs of chromosomes. So it's 2 similar genetic material, one from your mother, one from your father. So 2 genes that are the same, but they're different, right? One's from your mum, one's from your dad. So it's important to note as well in meiosis, we start off with diploid because we do have diploid cells, and we end up with haploid cells. So that's important to note. But the question here is asking us in a triploid organism. So that's organisms with not 1, not 2, but 3 copies of the chromosome. So in haploid, we have 1 copy. In diploid, we have 2 copies, which is shown here. And in triploid, we have 3 copies. So if we take a quick look here, the way this was drawn might confuse you. But it's important to just let's look at just one gene. Let's look at one gene. So 1 chromosome, 1 from the mum, 1 from the dad. Remember, this is diploid, so we don't confuse ourselves here. So during interphase, this might confuse you. So it's important to know the terms. What's a centrosome? What's a centromere? What's a chromatid? What's a chromosome? What's pairs of chromosomes? What's a DNA? What does all of this mean? So it's important to note that in interphase, we have here our chromosomes, but they have 1 chromatid. So this is a chromosome of 1 chromatid. It's not until it goes through interphase that it becomes a chromosome with 2 chromatids. So understand that a chromatid is a copy of a chromosome. So when he says how many copies of a chromosome, this chromosome here has 2 copies of a chromatid. So that's one important thing to understand. And it's joined here by a centriome. A centromere, not centriome, that doesn't exist. So a centromere, see, even I'm getting confused. So centromeres combine the 2 chromatids. So make sure you understand that. So over here we have 1 chromosome, but it's only got 1 chromatid. And that same chromosome has now replicated to become, it's still the same, it's a chromosome, but it's just got 2 chromatids. So they're just copies of each other. So if we just take a look here, you've got 1 chromosome from your mother or your father and 1 from your other parent. So let's say 1 dad, 1 mum. What happens is during interphase, they replicate. So you've still got 1 from your mother. So 1 from your mother, 1 from your father. And then during meiosis, so the first stage of meiosis, remember, so for prophase, the nuclear cell condenses, so it goes away, and then they pair up. So 1 from your father, 1 from your mother. So they pair up and they do this cross exchange of genetic material. And you remember in metaphase of meiosis 1, they go to the middle of the cell. So remember middle, metaphase, go to the middle, and then they're still stuck together. So the 2 pairs from your mother and your father, the 2 chromosomes are together. And then what happens is they split in anaphase, and in telophase and cytokinesis, you get these 2 daughter cells. But that's after first stage of meiosis. So it's in a diploid organism. What we see is that we have, if we're still focusing on 1 gene, so let's just say this is 1 gene, that's another gene. Let's just focus on 1 gene. So that's 1 gene. Remember, 1 from your mother, 1 from your father. So what we have here is 2 chromosomes, but because they're made up of chromatids, we have 1, 2, 3, 4. So we have 4 copies of our chromosomes. So remember, so that means we have 4 chromatids, so 4 copies of the chromosomes, even though you see here 2 chromosomes. So it's important to note. So when they're saying copies of chromosome, it's how many chromatids. So that's a diploid organism. Now for a triploid organism, it's not different because triploid organisms only have 1 extra It's only one extra chromosome. So it's not going to it's not going to affect the number of copies of the genetic material present. So therefore, if you just draw it in here, let's just draw it focusing on, let's just say 3 copies. What you're going to see is, if we just draw it here, so they're asking as well after meiosis 1, it's literally just going to be, here's your chromosome of 1 chromatid, and they just have to obviously replicate. It's going to be, here's 1 chromosome, 2 chromosome, 3 chromosome. They replicated, and you can see that now we have 1, 2, 3, 4, 5, 6. So 6 pairs or 6 copies. So 6 copies of your chromatids for 3 chromosomes here. So we have how many copies of your chromosome? 6. So that's your answer. So your answer here is 6. And it's just important to understand that I think a lot of students, from what I see from my time from teaching, is they get confused because they just get confused with the terms. So try to get an understanding of what chromosome actually is. And I think a lot of times we're taught how chromosomes change between different cycles or stages in the meiosis or mitosis. But I think it's important if you actually look at it as well from how are the chromatids changing, the chromatid number. That's probably a good exercise for you to do, because chromatid number is changing. It might look like the chromosome number is the same. You might say that there's one, so there's still 1 chromosome between this phase and this phase, but there's actually 1 chromatid and 2 chromatids in this phase. So that's one way to actually get your head around it. So try to understand what a centromere is, what's a chromatid, what's a chromatin, what's a chromosome, what's DNA, what are the special stages of meiosis? Obviously, meiosis 1 and 2, there's prophase, metaphase, anaphase, telophase. So there's 2 steps. The difference between mitosis, what's haploid, what's diploid. If you understand those terms, you'll be perfectly fine, and you'll be able to answer all of these with a breeze. So look, if you're still finding trouble, drop a comment down in the comment section. Love to help you. And if you do want to contact us privately, you definitely can. We'd love to help you as well. Thanks for your time. Bye now.